Good morning. Thank you all for coming. It's my pleasure to introduce today's Grand Round speaker, Dr. Keri Drake. Keri received her bachelor's degree from St. Mary's University of Minnesota and her medical degree from the University of Wisconsin School of Medicine. Keri was an outstanding pediatric resident at the University of Minnesota from 2010 to 2013. And since 2013, she's been a pediatric nephrology fellow at Cincinnati Children's Hospital and will complete her fellowship next spring. Keri has received numerous awards recognizing her research accomplishments, including the Shapiro Excellence in Student Research Award and the Midwest Society for Pediatric Research Best Student Presentation Award, both in 2007, research honors from the University of Wisconsin School of Medicine in 2010, the Pediatric Scientist Development Award, which supports protected research time during fellowship in 2013, the Arnold W. Strauss Fellow Award in 2015, and the Fellows Basic Research Award of the Society for Pediatric Research in 2016. Keri's topic this morning is Deciphering the Hox code in the developing kidney. Keri, we look forward to your presentation.
Thank you so much for the introduction, and it's really great to be here. So today I'm going to talk about some of the clinical and research interests that I've developed during my nephrology fellowship, and then I'm going to share with you the project that I've worked on during the pediatric scientist development time, looking at Hox genes in the developing kidney. So I have no disclosures, and I won't be discussing any off-label products today. So my objectives today are to give an overview of congenital anomalies of the kidney and urinary tract, and then I'm going to discuss some of the challenges in understanding the pathophysiology of these disorders and their relationship to basic science research in renal development. And then I'm excited to share with you my work in our Hox mutant mouse model that's shown some interesting phenotypes and provided some insight into the role of Hox genes in the developing kidney. So first, I'd just like to review congenital anomalies a little bit and talk about why I've become interested in renal development research. CAKUT anomalies are the most common cause of chronic kidney disease in children, and depending on the cohort studied, the incidence ranges from anywhere from 40 to 60%. And these malformations include renal agenesis, hypoplasia, dysplasia, causes of obstruction, including posterior urethral valves, and malformations like ectopic and horseshoe kidneys, and reflux as well. And as shown here, this accounts for a significant portion of chronic kidney disease in children. However, the natural course can be quite variable. Some patients have very mild, stable disease, where others are either affected severely since birth or go on to have progressive disease and end-stage renal disease. And that's reflected here in this graph where you can see the age at the start of renal replacement therapy in CAKUT patients. And you can see that a subset of patients require renal replacement therapy within the first few years of life. And then there's a peak around adolescence during a period of rapid growth when these patients sort of outgrow their residual renal function. And there's been significant research looking at the genetic causes of CAKUT. There's been over 20 genes identified in humans, and these genes have been identified through different approaches. Many have been found through hereditary syndromes that have renal and extrarenal malformations, and then the genetic cause can be identified through that approach. And another significant approach has been through looking at renal development in different biological models, such as a mouse model, in which knockouts produce a renal phenotype, and then these candidate genes can be examined in patient cohorts with CAKUT. And then with the advances in next-generation sequencing, novel mutations are being identified, and this list of 20-plus genes is constantly growing. However, the majority of patients with CAKUT are not found to have a known genetic mutation. Depending on the cohort, reports have ranged from 6% to 30% in identifying causative mutations. And this variability is likely due to several things. Different cohorts have looked at different targeted genes being sequenced. So depending on how many genes were examined in that cohort, that can make a difference in how many patients were identified with a known genetic cause. And then also the cohort that was selected. Some cohorts look at all CAKUT comers, where others look at more severe. And then whether or not the studies looked at familial data as well, since in the study that reported only 6%, these include family data, so that the study identified many genes that could potentially be pathogenic, but then these variants are also found in the normal unaffected parents, which rules it out as the sole monogenic cause for CAKUT. So I think that this starts to take a look at the complexity in trying to identify the genetic causes of these diseases. And then I just want to highlight that looking at this list, PAX2 and HNF1 beta are some of the more common variants identified in CAKUT cohorts. But you can see that mutations in these genes are associated with different forms of CAKUT. One thought that could be due to that these are big players in renal development and are likely involved in multiple stages of the developing kidney, and depending on the degree of function loss in the protein, that can make a difference in the phenotype that's observed in the patients. But again, this further highlights the complexity in trying to sort out the pathogenesis of these diseases. And then further adding to the complexity, there's evidence that epigenetics, modifier genes, gender all play a role in the manifestation of CAKUT. And then cannot forget to mention the environmental and teratogen effects as well. So maternal diabetes, nutrition, drug exposure, intrauterine growth restriction, all have shown to affect renal development and can result in congenital kidney anomalies as well. So I showed this slide just to kind of summarize the complexities in trying to understand the etiology in CAKUT. Here you can see that there's likely rare alleles causing monogenic disease, but this accounts for probably a very small proportion of the number of cases. And then there are more common alleles, either at low frequency or even maybe more common in the population, that would normally affect renal development, but the process is robust enough, or there's modifier genes or a second gene in a similar pathway that then causes some patients to be affected where others are able to have normal kidney development. So in that context, I just wanted to highlight, while I've developed the bias and think that renal development research is so important, it's not only fascinating from the perspective of understanding how a complex organ like the kidney can form, and also providing those insights into the genetic pathways involved in congenital kidney anomalies and nephron number, like I've mentioned earlier. But it's also fascinating from the aspect that many of the genes involved in development are again players that are re-expressed during injury and regeneration and healing. And the insights that can be gained from their role in development can then be applied to their function in regeneration and repair. And then excitingly, the mechanisms that have been uncovered in basic renal development are being applied to the advances in organoid generation from pluripotent stem cells. And the field is really making significant advances in that. So that, the further that we understand the basics of renal development, I think that will continue to push the organoid and organ regeneration field forward as well. So next, I'd like to spend a few minutes just reviewing the basics of renal development. And first, understanding the very early stages in development is not only fascinating from a basic developmental biology perspective, but has also been exciting to me in the fact that I think it has implications in our research project. So very early in development, there's precursor kidney structures that develop, and this includes the pronephros, which is a single non-functioning nephron in mammals that degenerates, and then is followed by mesonephros development, shown here. This includes several nephrons with a single tubule that connects to the nephric duct. And while this remains the functional kidney in fish and amphibians, in mammals, it either degenerates in the female, or in the male differentiates into parts of the reproductive tract, including epididymis tubules. And then following the mesonephros, the metanephric kidney develops, and this is the structure that becomes the functional kidney in mammals. So taking a closer look at metanephric kidney development, you can see here that the uteric bud branches out from the nephric duct, and that is in response to the metanephric mesenchyme, which are these nephron progenitor cells. And then through development, the uteric bud undergoes secondary branching in crosstalk with this cap mesenchyme or metanephric mesenchyme. that then gives rise to the parts of the nephron from the podocytes, glomerulus, through the proximal tubule, loop of Henle, and distal tubule, whereas in the mature kidney, the collecting duct arises from the uteric bud lineage.
Is this mouse the E11?
Yes, so this time points here are in mice. So at E11 is when the uteric bud branches out to start with metanephric kidney development. And then birth for the mouse normally occurs around E20, but they continue to form nephrons all the way until postnatal day 2 or 3. Whereas in humans, this timeline corresponds to about the 5th week of gestation, shown here. So the pronephros, the mesonephros form very early and degenerate early in development, whereas human nephrogenesis continues to about 4 to 36 weeks gestation, and then no more nephrons are formed after that. So using the mouse model, CAKUT-associated genes have been identified and validated as in humans, as shown in this diagram. And you can see different CAKUT-associated genes in the mouse here and in the human here correspond to different stages within development, as shown here with uteric bud induction very early in metanephric kidney formation, with genes identified that are important in mesenchyme to epithelial transition, as well as branching morphogenesis, and a few genes that relate to the nephron patterning and nephron elongation. And many, but not all, genes that have been associated with CAKUT anomalies correspond to the different stages in development. So there are a few that still remain to be further characterized. And then it's interesting to note that most of the genes that have been identified so far are related to this very early metanephric kidney formation and UB crosstalk with the metanephric mesenchyme. So next, I'd like to switch gears a little bit and talk about my specific research examining the roles of Hox genes in renal development. So Hox genes have long fascinated developmental biologists. Over 100 years ago, homeotic transformations were discovered in fruit flies, and this is where one body part is replaced by another. As you can see in the fly examples here, that either develop an extra set of wings or have antennas replaced by legs. And eventually it was discovered that these mutations were secondary to Hox genes, and the Nobel Prize was actually awarded to the scientists that described how Hox genes confer segment identity. So Hox genes function as master transcription factors in development. They contain a homeobox that corresponds with a homeodomain to bind to DNA, and they can either activate gene expression or repress gene expression depending on the context and cofactor binding in the different sites of the protein as well. Hox genes are highly conserved throughout evolution. You can see here that more primitive species have just a few Hox genes that were duplicated throughout evolution to a total of 39 in humans and mammals. And this was thought to occur through evolutionary duplication, in which one ancestral gene is duplicated, takes on additional function, or it provides a survival advantage, and gives rise to more and more Hox genes, again resulting in a total of 39 in mice and humans. Hox genes are best known for their function in determining anterior-posterior patterning of the developing embryo, and this has been well characterized in the axial skeleton as well as the limb shown here. And then another fascinating property of Hox genes is that they show spatial collinearity. So this means that the order that they are located on the chromosome corresponds to the order that they are expressed within the anterior-posterior patterning of the embryo, as shown here in the fruit fly anterior-posterior body patterning, as well as in the developing embryo. And then this is the classic kind of structural representation of Hox genes in mammals, and they are arranged by sequence homology to correspond to the 8 Hox genes found in Drosophila. And as you can see here, Hox genes are arranged in kind of a vertical alignment that are designated as paralogous genes and are thought to have arisen from a common ancestral gene, and then also flanking genes, which are arranged horizontally here. Given their mechanism of duplication throughout evolution, there's a significant amount of functional redundancy within the Hox gene network, and this is thought to be strongest in the paralogous group, but is also seen among related flanking genes. So that the loss of one Hox gene produces little to no phenotype, whereas the loss of a whole group can cause significant developmental defects. However, as beautiful this diagram is in its color coding, it's clear that the function of Hox genes do not follow the distinct color-coded borders. So really the central challenge in studying mammalian Hox gene is dissecting out where the functional redundancies overlap and where the distinct functions are seen. And then I just want to take a moment to talk about what's known about Hox genes in human disease. It was generally thought that Hox genes are so critical to development that any loss of significant number of Hox genes would result in non-survival, and therefore we wouldn't necessarily see a proportion of disease related to Hox genes in humans. However, there have been germline mutations in 10 Hox genes that have been associated with human disorders. However, there's significant variance in penetrance, inheritance, and expressivity. So even the same gene and very similar truncating mutations can be associated with very different phenotypes. And this includes phenotypes in the CNS, the limbs and extremities, cardiac phenotypes, and genital abnormalities as well. But again, there's quite a bit of variation in these rare syndromes that have been identified. And then there's also human disorders associated with Hox gene regulators, such as ASXL1. And then Hox genes have been shown to play a role in different malignancies. And it's thought that they play a significant role since Hox gene deregulation can be associated with prognosis and aggressiveness of the cancer. However, it's also not clear exactly what that role is. The same Hox gene in one cancer can be upregulated, where cancer in a different tumor, tissue, that same Hox gene is downregulated. So it appears that this Hox gene signature it differs among different tissues and likely is not easy to sort out what that role in cancer is. So next I'd like to talk about what's known about Hox genes in renal development. Work from the Potter lab has shown that nearly all 39 Hox genes are expressed during development, and they're seen really in all components of the developing kidney. This includes the uteric bud that branches during development and forms the collecting duct, the cap mesenchyme cells, which are the nephron progenitor lineage, and then the stroma or support cells around the developing nephron structures as well. Studies have looked at Hox10 paralogous mutants, and these mutants develop renal hypoplasia with abnormal uteric bud branching, as well as Hox11 mutants. In these mutants, the kidneys fail to form at all, and it's thought to be due to lack of the uteric bud to form in communication with the metanephric mesenchyme. And then also HOXD cluster mutants, where they've knocked out different combinations of the HOXD flanking genes, develop renal hypoplasia, and they see a few dilated tubules and a few scattered cysts, but a relatively mild phenotype associated with the loss of the HOXD genes. So the central hypothesis of our study is that the roles of Hox genes in these previous studies have been concealed by functional redundancies. And through a genetic dissection of different flanking and paralogous genes, we hope to uncover novel Hox function within the developing kidney. So next I'd like to talk about the method that we've used to try to get at this and the phenotypes that we see in our Hox mouse model. So to generate the mouse lines, the Potter lab knocked out specifically combinations that relate to the abdominal B cluster genes. So this includes Hox A, 9, 10, 11, C, 9, 10, 11, and D, 9, 10, 11. And I'd just like to take a minute to walk through this diagram and describe the unique recombinating strategy that the Potter lab has used to generate these mouse lines. They used a method to induce a frameshift mutation in each first exon of each individual Hox gene. And what's novel about this method is that it maintains the intergenic regions that can contain enhancers, so that it's a very clean dissection instead of knocking out a whole block of Hox genes. The intergenic regions contain enhancers that can then affect the expression of the remaining Hox genes. So that makes it difficult to know if the phenotype that you observe in that model is due to the loss of the Hox genes that you are specifically knocking out, or if it's due to misexpression of the remaining Hox genes because you've also lost those enhancer regions. So to do this, a plasmid with once only LOX-P sites was recombined into a BAC containing the Hox cluster, and each of these specialized LOX-P sequences carries a mutation so that the single remaining inactive LOX-P site does not interfere with subsequent modifications of the flanking genes. And then introduction of this LOX-P site results in a frameshift deletion and thereby knocks out the function of the remaining protein. And then mouse lines were generated using standard techniques in which the construct was electroporated into embryonic stem cells, and then using chimeric mice and through a series of selection, they're able to generate these mutant mouse lines. So the Potter Lab has generated lines of A9, 10, 11, C9, 10, 11, and D9, 10, 11, and then through breeding we're able to make these different combinations. So in our study we focused on wild type and single hets, which were used as controls, and then characterized different combinations of heterozygous, homozygous, triple hets, and double homozygous mice as the mutant combinations that we focused on. So first we looked at survival of these mice. Double homozygous mice were embryonic or early postnatally lethal with very few exceptions. And then heterozygous homozygous combinations as well as triple hets showed early mortality with about 75% survival at 2 months and about just over 50% survival at 4 months. To characterize the embryonic phenotypes, we did gross dissections at E18.5, so this is just prior to birth, and we have representative images shown here, where you can see the arrows depict the gonads, and the asterisks show the adrenal glands. And we see in triple hets and het homozygous combinations of AC and CD significantly smaller kidneys. And then in the double homozygotes, we often see renal agenesis, where the kidneys fail to develop and are not present at all at E18, or in some mice with some variable penetrance, we see very small, irregularly shaped kidneys that are present there at E18 as shown there. And we quantified this in all the genotypes shown here with asterisks showing statistically significant differences. And I just want to highlight again the heterozygous homozygous combinations where we see the most variability in the A knockout C hets. But again, the A het C knockouts, C knockout D hets, and C het D knockouts were severely smaller and compared to wild type and single hets. And then when kidneys were present in the double knockout mice, again, we see that they are significantly more affected than the het homozygous combinations. And then, interestingly, we see a trend towards smaller kidneys in the C knockout. The p-value here is 0.6, so that we think if we get a few more numbers that this might be statistically significant. But again, is quite a different observation than seen at least in the A homozygous mice, suggesting that the C homozygous mice Hox cluster may be playing a more significant role than previously appreciated. We next looked at histology of these, and in the het homozygous mice, the organization of the renal structure and the renal architecture looks normal. So this fits with a phenotype of renal hypoplasia. In the double homozygous mice, we see that the renal architecture is disorganized with increased stromal cells surrounding tubules consistent with peritubular cuffing that is suggestive of a phenotype of renal dysplasia. We also characterize the phenotypes observed in Hox mutant mice that survive to 2 months of age. And in comparison to wild type, we see a small percentage of mice that are relatively unaffected. And again, this isn't a big surprise to us, as our mice are on a mixed genetic background and we expect to have some variable penetrance. And then I just want to draw your attention to the group of mice that then are more severely affected, in which we observe either microscopic cystic disease or grossly cystic disease as well. And then we do see phenotypes in the middle of this with hydronephrosis. And we've quantified this and haven't seen a difference between the genotypes so far. But again, with our variable penetrance, it takes a large number of mice to really tease out if there's a difference in the genotypes causing the different phenotypes that we see. Given that we observed small kidneys, we wanted to quantify nephron number. We used the acid maceration technique, which is kind of a faster and dirtier method of determining nephron number. But because the kidneys were so dramatically small, so much smaller, we use this to be able to get a sense of also the variability that we see. And in the het homozygous combinations, as well as the C homozygous mice alone, we see a significant decrease in nephron number, with the most severely affected mice showing about a 60% reduction in nephron number. So using these models, we wanted to dig a little bit deeper and to see if we can get more information about exactly how Hox genes are functioning to produce these phenotypes. So first we looked at uteric bud branching early in development at E13.5. So this is just a couple days after the metanephric kidney is forming. And we see that in wild type kidneys, they have very symmetric branching, both in the anterior posterior axis and then dorsal ventral as well. However, in the heterozygous homozygous combinations, we see that branching either appears just in the dorsal axis without ventral branching, so a pretty significant asymmetry, and then a subset were more severely affected with branching just in the dorsal axis and just in the anterior axis. And then the double homozygous mice showed phenotypes with either these just a few elongated branches as shown here, or complete lack of branching with the representative images shown here. And we wanted to quantify this by the different genotypes. And interesting, we saw that the A het C homozygous were more severely affected, as well as the C homozygous D hets, again suggesting that the Hox-C cluster is playing an important role in this early branching. And then the A het C homozygous, A homozygous C homozygous mice most consistently had the decreased uteric bud branching, where we did see a few of these elongated structures in the CD homozygous knockouts. We also quantified the number of UB tips in these images, and we can see that not only is there asymmetry, but there's decreased number of uteric bud branches as well. So knowing that the uteric bud contributes to nephron number, we next wanted to look at the nephron progenitor cells. And at E15.5, we see that the cap mesenchyme looks fairly normal in the het homozygous mutants. We wanted to look at proliferation to see if there were any differences, and there appears to be decreased number of proliferating cells shown in green in the cap mesenchyme cells stained in red here. And we're in the process of quantifying this for the different genotypes to confirm that there is a significant decrease in proliferation in these het homozygous mice. However, in the double knockout mice, particularly in the AC mice, we see early loss of cap mesenchyme, as shown here with, even though the uteric bud branch is present, we have areas where the UB branching is absent and dropped out due to the decreased uteric bud branching phenotype that we've characterized. We see that there are decreased number of green cells here that label the cap mesenchyme. So in the double homozygous mice, this phenotype is much more dramatic. And we've looked earlier to further characterize this, and we think that it is more severely affecting the AC combinations as opposed to the CD knockouts. So as you can see here at E13.5, there's less deuteric blood branches and almost appears to be increased amount of cap mesenchyme around each branch. And interestingly, the cap mesenchyme seems to track down along the deuteric blood branch, which isn't seen in wild type. And then in the AC double homozygous mice, we see just a few positive 6-2 cells, and this stain here highlights a single nephron and stains the few podocytes, but again does not label any cap mesenchyme cells. So we think that that is one other difference between the genotypes that our study is fleshing out. So after characterizing different components that could contribute to the decreased nephron number, we next wanted to look at tubular segmentation in these mice. So at E18.5, shown here, we used a marker of proximal tubule, LTA, in green, and a marker of collecting ducts, DBA, shown red in here. And in wild-type kidneys, these are very specific markers where we don't see overlap of staining. However, in our mutant mice, we see multiple tubules that are positive for both, either in a pattern of alternating cells, as shown here, or as shown here with cells that seem to dual stain for both markers. And this is, becomes prominent when we look at the adult mice, that even in some non-dilated tubules, you see an overlap of collecting duct and proximal tubular markers, and it's particularly prominent in the tubules that are dilating or forming cysts. We're excited about that observation, so we threw a ton of markers on it to try to confirm this. We've used 4 different proximal tubular markers and 5 different collecting duct markers, with a representative image shown here of collecting duct markers with LTA, a proximal tubular marker, and you can see how these cysts are dual staining for both, with again some cells being positive for both segments of the nephron. And we confirm this with other proximal tubular markers, as well as 2 loop of Henle markers as well. So these cystic kidneys are showing a pretty significant misidentity of tubule cells. We often see the misidentity at E18.5 in scattered tubules, but rarely we've picked up a few mutants that have cysts at E18.5, and again with dramatic overlap of tubular segmentation. So we think that the small percentage of mice that go on to develop cysts in adulthood may be developing those cysts during development. And then to further support what we're seeing by immunofluorescence, we did scanning electron microscopy. So this is where the lumen of the tubule is fractured open. So you're looking down onto the surface of the cells shown here. And this was a severely affected het homozygous mutant with cysts. And you can see the tubule mainly appears to be composed of cells with short, sparse microvilli, but then has these regions of tightly packed brush border-like microvilli shown here. So the tubule appears to be consistent with a distal or collecting duct origin, but then has regions of brush border-like structures, which again is consistent with the overlapping of proximal and collecting duct markers that we see by immunofluorescence. So we're really fascinated by this misidentity of cells, especially because the proximal tubule collecting duct misexpression arises from cells that come from 2 different lineages. Because as you recall, the proximal tubule arises from the metanephric mesenchyme, and the collecting duct arises from the UB. And these 2 cell populations are specified very early in development, around E8 or E9. So I just want to review the patterning that happens early in development and the next steps that we took to try to understand the etiology of this tubular misidentity. So as shown here, the uteric bud arises from the intermediate mesoderm, shown here in the green color. And this also gives rise to the mesonephric tubules, as shown here. And as you recall, these mesonephric tubules either degenerate or become the epididymis in the male reproductive tract. And a fascinating property of the mesonephric tubules that's been described in the literature is that they express both markers of the brush border and the collecting duct. So to us, that gave a sense of could there be some connection related to that of what we're seeing in our tubular misidentity in our Hox mutant mice. And then the metanephric kidney arises from the uteric bud, from the intermediate mesoderm shown here, in communication with the metanephric mesenchyme. And again, this lineage is specified early in development. So to examine this in our mice, we looked at the Mesonephros at E13.5, and you can see here in wild type the mesonephric tubules extend down to about this level, but in our double homozygous mice, we see mesonephric tubules extending down to the location of where the metanephric kidney is developing. And when looking at this in higher magnification, you can see these tubules being incorporated into the region of the metanephric kidney, both in the AC and CD homozygous mice, whereas we don't see these tubules forming in control kidneys. And while we haven't identified a marker to say specifically differentiate between this is a mesonephric tubule versus a metanephric tubule, by structure and the differences that we see in the double homozygous mice compared to wild type, there seems to be some structural differences in tubular formation at this age. And then the uteric bud of the mesonephric kidney does not undergo secondary branching as we see in the metanephric kidney. And interestingly, as we've examined more of these mice, we see that the anterior branch of the uteric bud in this kidney fails to undergo secondary branching. So again, concerning for could there be some type of anteriorization or mesonephric-like properties occurring in our metanephric kidneys. To further support this, we did gene expression profiling of whole kidneys at E18.5, kind of as a global view to see what genes are differently expressed. We looked at A knockout, C knockout kidneys versus wild type, and as expected, we saw downregulations of genes involved in the uteric bud and in the cat mesenchyme, which is confirmed by what we showed with immunofluorescence. But then interestingly, we saw upregulation of genes involved in the extracellular matrix, and by gene ontology, genes that are consistent with being expressed in epididymis tubules with very significant p-values. So we wanted to confirm this by doing immunofluorescence, and we used 3 markers shown here of epididymis specific markers of RPS12, aquaporin 9, and claudin 11, which are found to be expressed in the epididymis, but you can see are not expressed in control kidneys. And these are upregulated in the adult mice cystic kidney tubules, as shown here. So we thought this was a pretty fascinating phenotype and wanted to see if there was any relation to human cystic kidney disease and what we're teasing out with our mice. So looking at human multicystic dysplastic kidneys, we rarely do see tubules that express both LTA and DBA, as shown here. And again, these markers are specific in the human control kidneys. And then we wanted to see if any of the markers from our mutant mice are also expressed in human MCDK samples. And interestingly, we do see expression DLK1, LOX, and decorin, which were identified from our RNA-seq data, are shown to be upregulated in human multicystic kidney samples. And then also 2 markers of epididymis tubules, including RPS12 and keratin 5, you can see here are expressed in the human MCDK samples and not expressed in controls. So for us, it's kind of an exciting finding. We never set out to have a translational component to our Hox project, but I think we've been able to identify at least a few markers that haven't been previously shown to play a role in human MCDK, and may provide an area for further studies. So in summary, our study has shown that there is significant functional redundancy of the flanking Hox genes, and highlight the importance of the HOX-C cluster. And then we've been able to further contribute to the phenotypes associated with Hox function in the uteric blood branching and role in nephron progenitor maintenance. And then excited about this novel phenotype that may represent a partial transformation with the metanephric kidney showing properties of the more anterior mesonephros. So for future directions, there are still further studies to really examine the early patterning of the intermediate mesoderm to help confirm that this is what we are seeing in our Hox mutants. And then we're also working on a project proposal to study the role of Hox genes in adult kidneys with the adult phenotypes that we have characterized. And with that, I'd just like to acknowledge the funding and mentorship through the Pediatric Scientist Development Program, as well as the Potter Lab, who Mike Adam has constructed the mice, and then all the members of the Potter Lab who have really helped me move forward with this project. And then the different cores at Cincinnati Children's between pathology, EM, and confocal microscopy, and really the support that they've given to help me move this project forward. And I'm happy to answer any questions.
The mesonephric tubules in both male and female kidneys?
We do. So we have female mice that are that develop cystic kidneys that are positive for those epididymis markers.
And the other question is, so the in the double mutants where you have loss of the 6-2 positive cat mesenchyme, so Tom Carroll's lab has shown that the proliferation, so the self-renewal of those stem cells, as well as their differentiation, is beta-catenin independent. Have you looked at Wnt signaling?
We haven't yet. No.
Wonderful talk, thank you. Maybe it was my imagination, but in some of your earlier histological figures, it seemed like there was some inflammation between some of the tubules. Do you think that there's a role for inflammation in part of the phenotype?
Yeah, so in the adult samples, we absolutely do see an infiltrate. We've reviewed this with the pathologist, and their feeling is, these mice have chronic kidney disease and have some tubular atrophy and loss. So their thought was that it's consistent with kind of an end-stage renal disease picture, and that's what we see when we look at the kidney function as a marker of BUN in these mice, that they're severely affected. So we think the role of inflammation is probably consistent with what is seen in chronic kidney disease, but Hox genes are important in really all cells, as well as inflammatory cells and the blood cell lineage. So there could be more there to that phenotype.
Can you speculate a little about what your future direction is in terms of the role of these genes in the adult kidney?
Yeah.
Beyond sort of the developmental patterning, what do you think is going on?
Yeah, absolutely. So initially I was, thrown by having this variable penetrance where some of these mice look completely normal. Histologically wise, the kidneys are indistinguishable from wild type. They have no proteinuria. And then BUN and creatinine are normal. I was doing electron microscopy on these mice, and fascinatingly, even in these normal appearing kidneys, we see increased autophagosomes and autolysosomes. Now, the epididymis-like properties of these tubules is something that has emerged relatively recently, and we haven't gone and validated all the markers that we have to see if they are expressed in these seemingly normal kidneys. But there's a paper that came out a couple years back showing that Hox genes repress autophagy. So our hypothesis is that the loss of the Hox genes in our mutant model is causing either upregulation or dysregulation of autophagy and accumulation of these autolysosomes, regardless of proteinuria or another form of renal disease. So while our Hox mutant mouse model is probably not the perfect model to tease that out, that's an area of study that we're really interested in, since Hox genes are expressed in the adult kidney and their function is not well understood. So we'd like to be the ones to show that Hox genes regulate autophagy in the kidney.
Nice job, Keri. It's great to see your work progress from your talk last spring at PAS, which was also excellent. Congratulations on the research award, by the way. So I wanted to ask you, your LOXP knockouts in these intergenic constructs, has anyone doing CRISPR-Cas9 mutagenesis to try to be a little more precise with where these specific SNPs might be?
Yeah, so we haven't. So the thought was, because these genes are so tightly linked and we've knocked out so many of them, that this would be sort of a challenging construct to make using CRISPR technology. So we haven't pursued that any further.
Is anybody doing deep sequencing of children that come into the clinic with ill-defined idiopathic cystic dysplastic kidneys to look at these homeobox genes, these Hox gene promoters? Is somebody doing that work?
So I know, people are looking at CAKUT in general, and I'm not sure in the cohorts how many multicystic dysplastic kidneys are there. We have thought about, do we try to pursue further, could Hox genes be leading to this mispatterning and then be leading to the development of multicystic dysplastic kidney? And we're haven't gone gung-ho on that yet. There's some challenges in working with Hox genes. There aren't great antibodies. The multicystic dysplastic kidneys are so heterogeneous that if you were to try to RNA-seq that, it's sort of comparing apples to oranges when you compare it to a normal kidney, because there's so much non-renal appearing tissue there. So, if we did see differences in expression in Hox genes, we don't know if that would be, how meaningful that would be. And then multicystic dysplastic kidneys can occur unilaterally. So could there be some patterning defect that is just occurring locally that may be related to Hox genes, or another mechanism downstream like PAX2 that's been associated with multicystic dysplastic kidney? So maybe our mechanism doesn't exactly translate to human, but I think that we're showing this mesonephric-like phenotype in multicystic dysplastic kidneys that hasn't been shown to this degree before.
Should I say it was a beautiful talk, like everybody else said? Yes, it's beautiful. I am intrigued, by the Hox genes and cancer as well. So, did you see any cancers in your models? Did you see?
We did not.
Any suggestion or?
Yeah.
No?
We did not, and we've looked pretty hard. We watch these mice pretty close and try to let some age and then catch them before they die on us and take a look at their kidneys. So, because we worry about, could these be developing later on if we just don't let the mice get old enough to see it. But of the work that we've done right now, we have not picked up any.
And based on your penultimate slide, I have an idea for a grant we will write together. So I work on a kidney. It's not a kidney disease; it's a skin disease, epidermal bullosa. 30% of these individuals develop renal failure, and the keratin 5, is the main driver of one of the subgroups of the EB simplex. So they're almost, I'm gonna investigate more and then we'll touch base.
Awesome, thank you.
See?
So, as you know, children with renal dysplasia often display a urinary concentrating defect. They're polyuric. And although I've observed that, everyone has, I never really thought very deeply about why that's the case. But do you think it's possible that this misidentity of cells is part of the underlying, could be part of the underlying pathophysiology of urinary concentrating defect in dysplastic kidneys?
Yeah, and that's something that we've thought about and taken just a few steps to try to work out, and then just haven't pushed forward. So we tried to look initially for a urinating concentrating defect in these mice, but there's a fair amount of variability in our accuracy in measuring urine output. I think in my first pass at trying this wasn't as good as maybe could be, and especially since we see hydronephrosis in these mice. One hypothesis is, could that be a functional hydronephrosis, that they're producing so much urine that then the system isn't able to drain, because we don't see ureter abnormalities. So the other piece that we think is really fascinating to this is that in the collecting duct, some of those cells that I showed that kind of have that alternating pattern, even in non-cystic mice, seem to label with LTA more strongly as a proximal tubular marker. In normal kidneys, there's very weak LTA expression, but in these cystic mice, it's pretty, and even some of the non-cystic mice, it's pretty dramatic that you see this increased LTA expression within the collecting duct. And could there be some abnormality? We see aquaporin expression just structurally that looks pretty normal, but could there be some functional defect there? And as I've been learning more about functions of the epididymis tubules, that they have intercalated-like cells that are important in acid base and water transport as well. So I think that there's also something there that might be able to be explored further.
Are there any other questions for Keri? If not, thanks for a fascinating talk.
You have been